Good afternoon, everyone. My name is Pavan Suresh, a 16-year old grade 11 student. And you may be wondering why I'm up here to present to you guys today. Well, just like every other presenter who was there earlier, I have my own story. And mine begins 10 years ago when I lost my great grandma to a tumor related cancer. And honestly, even though I was 6 years old, I still felt that so much. And and her loss created a scar that was deeper than any blade. And then recently, at the start of my grade 11 year, I began to research what really causes these tumor related cancers. And I found out that the malfunctioning of the VHL protein is a crucial factor in what causes the tumors to multiply exponentially. Well, just imagine living with a biological footprint that contains a tiny, almost invisible typo. So just imagine one protein, one cell malfunctioning causes this huge cancer. Well, similarly, for roughly 1 in 36,000 people, this isn't a metaphor. It's reality known as von Hippel-Lindau disease. It's a rare genetic condition, but its impacts are profound, causing tumors to grow in the body's most critical organs because the cells lose their ability to sense oxygen properly. But to fix this machine, I first had to understand what's already being done to treat it. And I found out the $75,000 treatment of Belzutifan. To a chemist, this is the perfect compound. It's everything in one. However, in the body, it does something else. After receiving the Belzutifan treatment, many people experience things such as anemia, which means low hemoglobin, so they have to import blood constantly, hypoxia, the feeling of low oxygen, headaches, and risk to the reproductive system. Well, all these now it's a no-go because these are all very harmful risks. So I thought, how could I cure this without using without using the medicine? And I found out using benchling for codon optimization. Well, what is codon optimization? Codon optimization is basically changing the spelling of the DNA sequence. So let's think of the DNA as a long strand of sentences going throughout. All I've done is I've changed certain parts so that instead of CAG, it would be CGA. And this is crucial because these codons are what gets sent to the protein in the tRNA to then get translated into the proteins itself. Well, enough of me talking about the codon optimization. I'll talk to you guys about what codon sequences I've changed. So I've changed places at amino acid 20, 38, 50, 62, 77, 96, 130, and 135. Well, at amino acid 20, there was a second methionine. And methionine, I like to call it as like the starting of a factory. So when you turn on a factory, all the things have to run. Similarly, methionine is responsible to start the protein translation in a gene. But however, the presence of a second methionine is really, really harmful for the protein itself. And instead, I changed it here to lysine because it creates a stronger bond. And then at amino acid 38 and 77, I changed the serines, which are both there, into glycine. And this is really important because phosphorylation in a protein is basically like a factory's whole energy being torn apart to its roots. So in a protein, if it gets phosphorylated, it stops working completely, causing bad things to occur. And then at amino acid 50, 62, 96, 130, and 135, I did something known as codon degeneracy. So let's imagine there's a factory and it's creating toys. Well, the machine that's creating the toys is doing it at a constant rate 24/7. So to make sure that the machine doesn't malfunction, they make the same toy again using the same codon sequence. So similarly, in the gene, we use the same codon sequence to help strengthen the protein and ensure no mistranslations occur. Well, all this I talked about, and what's next? So basically, I have the blueprint, I have everything now, but all that's left is to prove it. And I'm going to prove it using a machine learning model, using PyTorch, BioPython, and many other tools to help prove that this is a strong hypothesis. Thank you.
Perfect. Thank you so much for your
Yeah.
talk. It was very inspirational, obviously, in terms of medicine very detailed into the specific amino acid sequences. So it was obviously very inspiring there.
Yeah, and I think it really shed light into what can actually happen if you
Yeah.
get computational power and really apply that to
Yeah.
medicinal structures, like actually predicting where a protein fold is going to happen and like where exactly to target drugs inside of a protein as well and things like that. So I think with that we can open the floor to some questions.
Does anyone have any questions they would like to ask?
I think we'll give it a second because it was a pretty in-depth talk.
Okay. Can give it a few minutes, maybe.
Well, do you guys have any questions for me? Like you, Kevin, and Arlen? Do you have any questions?
Oh,
I think
personally,
Inspire X has a question actually.
Yeah.
Can you pin it?
oh, okay.
Yes. So
More of a
Inspire
general
X is
question, I
asking,
guess.
how does protein structure determine its functions?
Okay. So proteins, like I might have to get a bit technical here because I can't use the basic knowledge because I kind of like lowered the technicality during my presentation. But basically, proteins, when they get translated, they get folded into different structures. There's primary, secondary, tertiary, and quaternary, right? So when they get folded, they all form a very specific shape. It's like a fingerprint, for example. No two finger, like no, like Coven's fingerprint is not the same as mine. Similarly, the proteins are not the same. They all have a specific shape. So basically, like the VHL protein, it's like really flexible because there's some things called substrates and ligands, which are like factors that lead to cancer, right? They bind and they degrade. So like the structure, if there's like more binding sites and many things like that, it helps run more efficiently. So basically, I have hypothesized creating a more efficient protein structure. Basically, yeah. If that answers your question, if you want, I could explain more to you. Yeah,
Yeah, I
I
think
think that
that might
makes
have
sense.
answered
Yes.
his
Yeah.
question. Yeah.
Aside from that, I also just obviously for every single talk, speakers talk about their experience bringing them to their topic, or someone in the chat asked them about their background leading up to it. So, what really, obviously, you talked about your, I think, great grandmother, great grandmother. Was there anything else leading you to more of the medical field or the
Uh,
biomedicine research field?
so what led me to this was like really I wanted to learn more because I was going into grade 11 that time, right? I was like I want to like get into like one field like well I want to like learn, right? So I decided like genes were pretty interesting, right? Because I finished biology during the summer that time. So I was like, yeah, I want to do it. And like yeah, like right now I'm working with a few profs to get like this confirmed, right? So yeah, that's basically it. Just wanted to learn.
It sounds amazing. Yeah.
Yeah.
Oh,
Yeah.
we have another
Also,
question from Ethan.
yeah,
Okay. Yeah.
Ethan has a question.
Okay.
Okay.
I mean,
Um, Yeah, in the field of biomedicine, how do you think your work can shape it in around 5 to 10 years,
Yeah.
and how could that look?
Ethan, that's an amazing question. Right. So like this field, right? What I'm doing is called computational biology. So it's basically taking these genes and you're like using like different AI ML models to like help model the stuff, right? So like usually this research to a person that has a PhD, it's like their whole life, 30 years, everything. But like the VHL protein, as of now, right? It's like a technically all cancers have this because like like at least tumors because those tumors constantly multiply, right? So at the same time, the VHL protein is also malfunctioning. But now, because as I told you, like the earlier cure, which was Belzutifan, is not as good. So what I'm trying to do is I've enhanced the protein sequence, right? So that I can I can put in that sequence instead of the mutated sequence and switch it out, so that when that's not there, the normal one works better, kind of like that. So it can like help change the world of medicine for that.
yeah,
Perfect.
I think that's great.
I
Yeah,
think we
there's
have
one.
another question. I
Yeah,
think that's Amy Nanji, an early speaker today. I hope I'm right.
yeah.
Um, how do changes in a protein's three-dimensional structure affect
Yeah.
its ability to bind to other molecules and carry out its biological role?
Okay, so this goes back to the substrates and ligands, right? I told you guys earlier about primary, secondary, tertiary, and quaternary, right? Those are the four types of structures. And basically in proteins, right? Proteins are not just like circular, like a polypeptide chain. There's like many bonds occurring. Like there's the hydrogen bond, salt bridges, right? There's a lot of stuff going on. So basically, if you change something like in VHL, proline is like it's like really important for the degradation because basically these substrates and ligands they like sense the residue. So when they come towards that, it like binds. And for example, like if you change, like usually if you change like one protein in like something like sickle cell, like the sickle cell gene, it changes the whole thing, causing a mutation. However, like during the three-dimensional structure, when these bind, they have to degrade because this is in the normal conditions. But in the but then say it's not folded well. This is called like it's like misfolding. It's getting like it can't fold correctly because of these changes, right? So the three-dimensional protein structure being strong is crucial for this reason, and its biological role is very important. Like for VHL, it's very sensitive because it has to go around everywhere, right? Because it's degrading all these different things, so it has to make sure that it's doing its job correctly.
Yeah, so actually I have a question for myself too. So I
Yeah,
was actually wondering how like a protein structure can actually affect its oncogenic activity. Like for example, like some proteins might have a high content of like alpha helix. Like I think that some of the fork-head box proteins of the
yeah,
FOXO4 category usually do have like a high alpha helix content. So I was wondering how it like exactly would impact
yeah.
the oncogenic potential.
All right. So I'll be honest. I've never heard of the word oncogenic. Oh no, I mean like you mean
Oh
like
yeah,
oncology.
cancerous, yeah. Yeah,
Yeah, yeah,
cancerous, yeah.
oncology. Yeah, cancer, right? So
Yeah.
basically, right? VHL, it has like a bunch of like it's there's exon 1, 2, and 3, right? There's different exons which are responsible, right? So an exon 1 is basically alpha helices and beta splice. So it's just 2D folding because it's like a bunch of hydrogen bonds like all together in one, right? But then once you
Yeah.
get to exon 2 and exon 3, that's where the main thing occurs because that's where in exon 2 it's the most hydrophobic of it all, right? So it basically brings them together. It's like a locator, and then the exon 3 it brings it and then in exon 3 it binds it. I mean it degrades it altogether. So like what I'm doing, right, is like say you have the mutated like someone has the mutated gene or the
Yeah.
mutated protein, right? I'm just like
Mhm.
replacing it. So like because this has not been done before, like my civic sequence, and like I've already been continuing to build this ML model, right? So
Yeah.
it's like showing good results so far. So I hope to take this further. Yeah.
Yeah. So I think you're like trying to say like structure equals function basically, right?
Yeah, structure equals function because this one there's something known as gyration radius. It's
Yeah.
like from the center to the outer part. If it's smaller, it's more compact like that. So there's a lot of technical terms along with the
Yeah.
like the
Yeah.
grade you
Okay.
score. Yeah, hydrophobicity.
Yeah. Thank you. Thank you.
Yeah,
That was a really good explanation. So oh yeah, we have another question from
yeah.
Multier, I think. He's asking in terms of proteomics in general, is there a way to do this work more generally instead of too specific to specific amino acids and structures and find systems that make a more general solution?
Okay, so here's the thing. Like as I talked about with the medicine, right? That's used for this. Like a lot of these medicines come with side effects because they bind onto different things which can cause you like for example anemia, which means like the low concentration of hemoglobin, which is blood, right?
Yeah.
So like recently what I've been seeing, right, is like they've been doing something called personalized medicine, which is basically putting in like a enhanced version of the gene which is already there into the human body of someone who doesn't have like who has the mutation. So they're basically doing a switch, right? So for like and to answer your question, right, on the specific amino acids, I think I think you have to like look at the amino acid structures. Look, it's not for all of them. It's for this one because for this everything degrades through the protein. So for this one, I have to look at the specific to specific amino acid and structures. There could be systems in the future, but as of right now, I think it's very hard to find the systems. So therefore, it's much easier to look at the gene itself, gene protein, looking at the core cause of what causes all this.
Yeah. I think before you leave, I just want to ask you, have you heard of like Google's AlphaFold structures?
Yeah. Oh yeah.
Yes.
Actually,
Yeah,
I did
exactly. Yeah.
use that. I did use that for my thing. So I got the CIF file from that, and I used it to build my ML model. Yeah.
Yeah, exactly. I think that can really revolutionize some of this technology you're
Yeah,
talking about too, because it
yeah,
could actually like predict what it's gonna be, like how it's gonna fold, and that's really
yeah,
gonna tell you like which sites to actually target
yeah,
your drug bindings at, like a
yeah,
high kinase site, for
yeah.
example. But yeah,
Like,
thank you. It was a very
yeah.
insightful talk.
Thank you.
I wanted to quickly add one point. Um, I know at the end you've mentioned your machine learning model. I wanted to see, number one, if you had any progress, and number two,
Yeah,
just any general ideas you were thinking about it.
yeah, yeah,
Because we've, at least me and Arnav, we've been working
Yeah,
with health risk analysis and multi-layer health intelligence to integrate into the EnviroCast systems, especially in terms of like things like asthma and pre-existing conditions. But we've been looking at quantum accelerated proteomics and things like that to try and expand the suite. So we were saying, if you've obviously ML step one, if you consider different AI and even quantum computing for that.
yeah. Oh, okay. So right now, like I've heard of quantum computing, right? Because I think this one, IBM's made the quantum computer, right?
yeah,
So for
exactly.
yeah, IBM's made the quantum computer, right? So like, there's different tools. I mainly use like PyTorch, BioPython, right, and OS, like just to make sure it like runs properly. Because there's
Yeah,
different tools like OpenMM, right, which actually show you the molecular dynamics and everything. However,
yeah,
because now like a lot of the GPUs, right, or whatever, like the chips, they can't handle that much information. Because when I was using like the earlier one, I was like doing like the original drafts for my model. I was trying to use OpenMM, but it would crash every time because it needs like a NVIDIA core to run it. So I
yeah,
feel like when you're looking at quantum computing and this, like because right now like the universities are doing it well, right? I
yeah,
understand
exactly.
like they have it properly, but then I feel like for it to come to the human population, right, like to the general public, I feel like that's gonna take a bit of time because they have to increase the CPU cores because most computers right now are run on the Intel cores, right?
Yeah,
You probably will need like an NVIDIA core instead of that. So
yeah, like
and
the
that's a really good
4090
idea.
at minimum. Yeah,
Yeah,
exactly.
it's really unique
Yeah,
what you guys are doing. Yeah, and Enviro.
thank you, thank you. You
Yeah,
too, actually. Yeah,
yeah,
Thank you so much. Your talk was
thank
amazing.
you.
I really like how you really delved into depth. Your intelligence and experience with the subject really shows. Once again, thank you so much for doing this and sharing your work with us.
yeah.
Yeah, have a great
All
day.
right. Same to you. Bye.
There is always an opportunity to contribute to the discussions that we have at EnviroCast GEO. If you are interested in attending or even speaking at a future conference, learn more at geo.envirocast.org. We're excited to hear your insights.